section we're entering is about general neuroanatomy. Having covered the basic neuron and the three fundamental anatomical components of the neuron, we developed how a single individual cell works. Then we talked about how one cell is connected to another cell. And if you can connect two cells together, you can then string them together to produce pathways. Now we're going to go into the broadest component of anatomical description, and that's going to be the two main branches of the nervous system. Those two branches are the central nervous system and the peripheral nervous system. The difference between the two are easy to remember because the central nervous system, which sounds more important because it's called central rather than peripheral, involves only two components, your brain and spinal cord. Everything else is peripheral nervous system, and that makes it very easy. Brain and spinal cord versus everything else. And the brain and spinal cord, of course, because they're right next to each other, and they also have a fairly common property of not repairing themselves very well after damage, typically in adults, it's easy to keep that categorized well in your head. Now, the cells in the central nervous system involve some portion of it which will rewire itself in terms of neuroplasticity. So, for example, we were previously talking about the interneurons and how the interneurons are primarily the portion of cells inside the brain that rewire themselves so that you can generate new patterns of behavior. And that exists only in the brain itself, not in the spinal cord. The spinal cord does not rewire itself. And as I've mentioned previously as well, the peripheral nervous system, which is really made up of the sensory input pathways and the motor output pathways, do not rewire themselves at all. So of your entire nervous system, it's only the interneurons that exist in the brain that actually rewire themselves to generate new pathways. Everything else remains static throughout your entire life. Now, the brain and the spinal cord themselves are also categorized together in part because neither one of them repair damage very well that they receive. Brain damage and spinal cord damage should be fairly obvious to you to be permanent forms of damage. When somebody has a stroke or has a head injury, the brain may not repair itself well. Spinal cord injuries commonly give people lifetime problems. The peripheral nervous system, on the other hand, does involve cells that repair themselves. They are most easily seen, as I've mentioned before. If you chopped off your hand or your leg, you could go to the hospital and have it sewn back on, and then you'll be able to use your hand or your leg again. And that is because peripheral nervous system cells will clear away a dead cell and then generate a new cell to replace it. So if your hand were cut off and a doctor lined up the nerves in your wrist and your hand and essentially sewed your hand back on, the cells in the wrist and the hand will detect each other, clear out any dead material between them, and then form a bridge. Once that bridge is formed in your wrist, your brain can then send a signal all the way to your fingers, right, through the wrist where your hand had been severed, and make it all the way to your fingers. And then touch receptors in your fingers can then send signals through your hand across that new bridge in your wrist all the way up to your brain. That doesn't generally happen in the central nervous system. In fact, at best, if there is damage in the central nervous system, maybe 5 to 10% of that damage might be naturally corrected by the body itself, but most of the damage actually remains. One of the other issues about the nervous system that butts up against a common misconception is about how much your brain you actually use. The simple answer is all of your nervous system is available to be used all the time. Or as I've written here, 100% of the nervous system is working 100% of the time. That does not mean that every single cell has an action potential in it at every single moment in time. but no portion of the brain actually shuts down and is unable to process information from other areas of the brain. That's simply not true. And you have the basic rules to understand that. As we've gone through the function of a single individual neuron, you know that if positively charged particles get into the dendrite, run up into the soma, you cross your threshold, generate an action potential. When the action potential reaches the end of the axon, you have chemical neurotransmission from one cell to the next, and the whole process repeats itself in the next cell down the line. There's no process by which large vicinities of your brain simply shut down and don't do anything. So this old idea that you only use 10% of your brain is actually a faulty idea. And in fact, psychological historians have actually traced where it came from. A popular psychologist named William James, back at around 1900, was being interviewed by a newspaper reporter. And William James made the general statement that it may be that now in 1900, we may find out human beings have much greater potential than we're already aware. And then the newspaper reporter went back to his office and wrote, William James says we only use 10% of our brain. And that's where the myth came from. And it had been copied and copied and copied, and now it's nearly 120 years later, and you still have the plot line of only using 10% of your brain or 20% of your brain being in movies. It's simply not true. There's no part of your nervous system that ever shuts down. 100% of your nervous system is available to do work 100% of the time. Whether you're awake or asleep, it doesn't matter. Your brain and the rest of your nervous system is always working. And this issue actually will come back to us many times throughout the semester, because we will be talking about in the chapter on sleep. Why is it that if you're asleep and unconscious, you might still be able to learn or process information from events occurring in the environment, even though you're unconscious? One of the reasons is going to be because your nervous system never turns off. You stop responding to stimuli during the time that you're unconscious, but your receptors are still receiving input from your environment and still sending it into the nervous system, and it gets processed regardless of the fact that you're unconscious. As we get into the brain now, which is where we ultimately reach our final conclusions in this chapter, we're going to start from the lower sections of the brain, the areas that come just off of the spinal cord. And this is largely because the lower sections of the brain tend to be the oldest sections. So from an evolutionary standpoint, if you evaluate the construction of the brain over time, you really have just the spinal cord in a much earlier form of organism. And the brain at one end of that spinal cord simply starts to grow and grow and grow and grow. And so that the outer surface of the brain tends to be the newest section that you see, and that the lower section tends to be the oldest portions of it. The thing about the brain, if you take a look at this image on the screen here, is that the original spinal cord, even in the process of your own development, when your brain starts to develop at about 28 days after conception, you have essentially a rudimentary spinal cord, at which point your brain begins to grow in the same direction as the spinal cord itself. And then as it grows longer and longer, it eventually mushroom caps back and swallows up the tip of the spinal cord that it originally come from, so that the end of the spinal cord that you had at 28 days post-conception is actually now in the very center of your brain. So the oldest section of your brain is actually in the very, very center. And what you'll actually see, and this is an important anatomical issue, when you want to go from the oldest areas of the brain to the newest areas of the brain, what you want to do is think about going from the very center core of your brain off to the outer portions of your brain in any direction. You can go forward, you can go backwards, you can go to the side, or you can go straight up. It doesn't matter. The real issue between old to new areas of the brain is actually center toward the outside. But it's not front to back or just up to down. It is center toward the outside. The center is always the oldest, and the closer you get to the outer surface of the brain, the newer the sections of the brain are. In fact, the brain in general, as a very general rule, has a hierarchical construction in that the outer surfaces of the brain tend to control the sections underneath of it. So if you can imagine this, and this is somewhat of a inaccuracy and a simplification, but it's okay for our class to think of the brain as having the outer surface control a surface underneath of it, and a surface that gets controlled underneath of that, and that surface controls what's underneath of that until you finally reach the very center of the brain. And that would be a reasonable way, not perfectly accurate, but a reasonable way to understand general neuroanatomy. Now, when you take a look at the brain stem that you see in this image is really the tip of the original spinal cord you had 28 days post-conception, except your brain has grown back and mushroom capped around and swallowed it up. So what's called the brain stem is actually a residual component of your spinal cord at a very early point in life. And it's called the brain stem because it still has that spinal cord shape. It's a tube. Now, the very first section of that tube that comes off of the spinal cord is called the medulla. And the medulla, like any other area of the brain, always has multiple functions. But we will simplify which functions exist in a particular area because to try to understand all the various functions that are affected by one area of the brain is just an exceedingly difficult job, and you don't even do that in graduate school. But the thing about the brain is it's highly integrated, and the same cells that are used for one circuit are also used for another circuit. So to say in a general sense that if you got into the brain and you damaged one single individual cell, it is very, very likely that you have affected at least two systems, because the same cell is going to be part of a circuit for multiple systems, never just one system. That's the degree of integration that you see in the brain. So we're going to simplify which areas of the brain do what. And one of the most relevant aspects in terms of the medulla is that it controls heart rate and breathing. And this is actually why, as you might imagine, if somebody had what's generically referred to as a broken neck. Now, you might imagine now, with everything we've been talking about, you can't really break your neck. You could break a vertebra that's in your neck, but the spinal cord itself is not something that could be broken so much because it's soft tissue. It could be crushed or it could be stretched so that the cells disconnect from each other. And then once they do that, they wind up dying. And so as a result, if somebody really had significant damage to the neck, which is generically referred to as a broken neck, the medulla can no longer exert its influence on the control of your heart rate and your breathing. And what will happen is the person will suffocate, right? Because your heart will continue to beat even after it's disconnected from your brain. It'll beat for some time and then it'll stop, but your lack of breathing will cause you to pass out from a lack of oxygen much sooner than that. The cerebellum, which is the Latin word for little brain, is actually this section in the back of the brain here that if you popped it out, looks literally like its own little brain itself, but it's tucked in the back. The cerebellum is most oftentimes associated with coordination and balance. And as I've mentioned, as the brain rewires itself due to environmental experiences, you might imagine that a section that's related to coordination and balance would probably have a lot more integration in it in a person that does something like gymnastics or other types of athletic activity that involves a lot of motor coordination. And that's exactly what you do see. In fact, there's a variety of ways in which you can see throughout the brain using brain scans that areas of the brain tend to be more integrated in the sections that get used a lot. So for an individual that practices a lot of coordination and balance, like a gymnast on a balance beam, that individual would have a highly integrated cerebellum, much more so relative to the rest of us. The reticular formation you can't really see on this particular figure, but the reticular formation is essentially a formation that looks somewhat like a net that runs through your brain stem, and it's involved in general alertness. Not alertness from you drinking too much coffee, but just the fact that you can be conscious and awake and alive versus unconscious and unresponsive. It's that very overarching general gross form of alertness that we're talking about. And as you might imagine, if a person had damage to the reticular formation from a stroke, or if they were clubbed in the back of the head by a baseball bat, it could put them into a permanent coma. Because if you damage the reticular formation, they would lose the ability to be alert. And because this is central nervous system tissue, it doesn't repair itself very well. So the coma that they get put in could very well be permanent. The thalamus. Now, the thalamus is the very tip of your original spinal cord. This is the very center of your brain. It's the oldest section of your brain. It's the first portion that ever grew off of your original spinal cord. And what the thalamus is a central location for all of your sensory input. For all the experiences you have visually and hearing and touch and smell and taste and so forth, the receptors that are part of the sensory input pathway will run to the very center of your brain as though the thalamus were like a central train yard. And then those signals will get redistributed to the outer surface of your brain in the appropriate location from where they came. So touch will get sent to one section of the outer surface of your brain, vision will get sent to another, motor coordination will be sent to another. There'll be other variations there, but the outer surface of the brain is where you're going to wind up finding the analysis for a lot of sensory input. But there's always that initial location where things get linked together. This is also a location that would be closely associated with the concept of synesthesia. Synesthesia, which is something you may be familiar with, is when a person has their sensory input pathways crossed, so that if a person is touched on their hand, they will then have a perceptual experience of maybe a color, like yellow. So they'll say things like, when my left hand is touched, I see yellow. And that seems really very odd. People will see words when people speak. So they hear words, but then they have an visual perception inside their head. These same sorts of things, of course, occur when you hallucinate. So taking LSD or other types of substances to purposely make yourself hallucinate actually operates in the area of the thalamus, increasing the degree of connection between those cells and causing this cross manifestation of sensory input, so that when a person's high on LSD or psilocybin, they will have these odd experiences, like a sound will produce a visual experience, and the color red will make you see a particular object that's not there. Up from the brain stem, we get to one of the areas that's going to come back to us many times over the semester, and that's the limbic system. Now, the limbic system, you have to be very aware of in this course. The limbic system has one general purpose for us in psychology, and that is it controls emotion. So whenever I talk about the limbic system, you should automatically think emotional responding. And the limbic system itself doesn't show up on this image very well, but it is a pair of ram's horns, two large curls in the very center of your head that start from the thalamus and loop around like a pair of ram's horns, one on the left side of your brain, one on the right side of your brain. And all of the emotions that you can experience reside along those horns. At the very end of the horn, you're going to find the hippocampus, which is an area that's going to be related to memory. Now consider this whole story regarding the limbic system. Every emotional experience you can have resides along this large horn, and it's there as a way for you to express an emotional state. But given the degree of integration in the brain, it's also there for you to be able to detect the same emotions in other people. For example, you have an area of your limbic system that allows you to feel happy. It's generically referred to as the reward center, and it's along the very top of the horn. Now, if you were to damage the reward center, not only would you lose the ability to feel happiness, but you would also lose the ability to detect happiness in other people. If you lost the ability to express anger, you would lose the ability to recognize what anger is in other individuals. So the same area of the limbic system that's used to express an emotion is needed to be able to detect emotion in other people. And that operates just based upon the direction in which input pathways and output pathways go into these general vicinities for analysis. But it's a broader story that we'll talk about throughout the course of the semester. But it's very important that you recognize the degree of integration in the limbic system. Each area is used to express an emotion and to detect the emotion in other people. And because 100% of your brain has the potential to be operating at any one time, and I pretty much can guarantee you that your limbic system is always active, you will then always have an emotional experience for every single event in your life. And it doesn't matter what it is. Math anxiety, happy to meet a friend, enjoying coffee, anticipating an argument, all of those things reflect an emotional state. And because your limbic system is always operating, you're always going to have an emotional component to every single thing that you do. In fact, it's literally impossible. If you have an intact limbic system, you will have to have an emotional experience for every single thing that you do. Just to play with the idea a little bit, there's one person that's supposed to not have any emotional states at all, and that is Mr. Spock from the original Star Trek. Mr. Spock is supposed to be pure logic, no emotions. Let's play the game that you took a human being and you could, with some advanced technique, physically remove the limbic system from the right and the left sides of the brain so that there's no limbic system whatsoever. Would that person become purely logical? Would they function like Mr. Spock? Well, the answer is absolutely no. In fact, what you'd wind up with is an individual that would need full-time, around-the-clock care at a nursing home. Because if you ask that person, do you want to eat? They wouldn't be motivated to answer you, and they wouldn't be motivated to eat. They would lay in their own waste. They would never get up. They would have no motivation to do anything, because motivation, when you want something like I'm hungry, I'm going to walk to the kitchen, I'm tired, I want to lay down, I'm antsy, I want to get out, take a walk, or do some exercise. You won't ever have that emotional state as the beginning of a pattern of behavior. And in fact, what you wind up seeing is very often much of your behavior is actually initiated by an emotional state, and then the logic actually comes afterward. Because, for example, if you feel hungry, you are then motivated to eat, then you apply some logic as to, what would I eat? Should I eat at home or should I go out to eat? Do I want to eat something really healthy, or do I want to just go whole hog and eat a pound of bacon by myself? It depends, right? You start questioning things afterward, but the initiation of the behavior starts from the emotion itself. And so a person with no limbic system, kind of like Mr. Spock from Star Trek, would actually not behave like a logical Mr. Spock. In fact, they would just be largely incapacitated, and they would have to be cared for all the time. Now, the reward center is certainly one of the special areas of your limbic system. We're not going to cover all the areas of the brain, and we're not going to cover all the sections of your limbic system as well. But we are going to cover the areas that come back to us over and over again throughout the semester. That way, we have a good amount of repetition so that you actually learn the stuff throughout the course of the semester. And certainly the reward center is one of the relevant places of your limbic system that we need to focus on. For you to feel good about anything, you have to have your reward system intact. And your reward center, although you may think of it as kind of a couple millimeters or a centimeter at the very top of your limbic system, most of these centers, when we talk about an emotional section of the limbic system, tend to be drawn out circuitry that kind of spreads itself through the limbic system itself. So it's not like you have integrated circuits or little squares or boxes where you can just kind of identify exactly where the reward center is, and the anxiety center and obsessive center and anything else that you might be thinking about. They tend to be kind of a circuit that runs through this large horn-shaped section of your brain. But the reward center, by and large, is along the top for the most part. And it's used for any kind of reward that you would experience. So a very common one, any kind of drug addiction. Drug addiction must involve the reward center. People do not take drugs that don't make them feel good. They become addicted to substances that make them feel really good. The reward from it is highly addictive, and so they continue to go back and take more and more drug over time. But even things like learned relations through social attention are processed through the reward center as well. So when you learn to feel good because somebody congratulates you for doing a good job, that is also processed by the exact same area of your brain that would process heroin addiction if you had a heroin addiction. It's also the same area of the brain that would process how much joy you would feel from seeing your worst enemy fall down a flight of steps. And these three things seem completely different. Drug addiction, being congratulated for doing a good job, and watching somebody you hate get hurt. But each one of them, if you feel good after each of these scenarios, is actually going to be processed by the same area of the brain. So the reward center itself is not changing. It's other circuitry in your brain that's processing the experience overall. And you're not going to find one select area of the brain that just does one thing that kind of makes you happy because your worst enemy fell down the steps, or makes you happy because your best friend said good job. You're going to see a very broad integrated set of circuits throughout the brain, which makes it very difficult to figure out precisely which area of the brain is actually doing what. One thing to be aware of currently, and this is just simply a current issue at this time and around 2020 is that the drug ecstasy, also known as MDMA, tends to destroy the cells in which it works. And this is just a happenstance of a particular drug that's commonly available now, but it's worth making note of this. Ecstasy, you might imagine, as a drug called ecstasy should make you feel good, and generally it does. It's a stimulant that has some hallucinogenic properties. It happens to have a molecular shape that when used over and over and over again in the reward center, which is of course where it works, slowly destroys the cells in which it works in. As a result, there are going to be reductions in dopamine and serotonin, which are two neurotransmitters that are closely related to reward. And so as you slowly kill those cells off in your brain, you have less and less of an ability to get high from ecstasy, and also less and less of an ability to naturally feel happy. So oftentimes what happens with people that use a lot of ecstasy over a long time, they have a great deal of difficulty getting high off of ecstasy after a while, mainly because they kill off cells in the reward center. Because they can't get high from ecstasy anymore, they stop using it. But now that they're not using the ecstasy, they actually have a smaller reward center, which causes them to be more likely depressed. And this, from a social standpoint, may seem rather odd because the person will think, why aren't I happy? I stopped using drugs. I should be better. I should be happier. I'm moving on to something more important than drug use. But instead, they wind up with a low-grade permanent depression because you're talking about central nervous system tissue. It doesn't repair itself very well. And because the reward center has now shrunk in size because you killed off some of the cells, you wind up with more tendency to become depressed or melancholy. And oftentimes, people that use a lot of ecstasy over a long period of time wind up taking antidepressant medication afterward as a result, simply because they need to increase the activity of the reward center just to function at a normal level. Now let's take a look at this question. Damaging an area of the limbic system that controls emotional expression will also reduce your ability to detect emotion as well. And the correct answer there is absolutely yes. That's true. When you damage an area of the limbic system that controls emotional expression, it will influence your ability to detect the same emotion in other people, because the same area of the brain is used to express a behavior and is oftentimes used to detect that behavior in other individuals. That's part of the high degree of integration that exists in the brain. Now we move on to the cortex, the outer surface of the brain. The outer surface of the brain is the newest section of the brain, and the word cortex is Latin for bark, like the bark of a tree. It's the outer surface of the tree. Now, the outer surface of your brain has 4 general vicinities, and they're right next to each other, of course, but they're called lobes. 4 lobes of the brain. They're just 4 vicinities. And each of these 4 lobes have different general purposes. There's the parietal, occipital, temporal, and frontal lobe. And you'll definitely need to know each of these 4 lobes on our exams and to be able to understand future chapters this semester. The parietal lobe primarily analyzes sensory input from touch and from balance. The occipital lobe processes vision. The temporal lobe primarily analyzes hearing, that is auditory, and gustatory and olfactory input. So gustatory is sensory experience from your tongue, and olfactory is, of course, your ability to smell. And then your frontal lobe is the only one of the 4 lobes that's used for motor output. So of the 4 lobes, 3 are dedicated to sensory input, and only 1 is dedicated to motor output. And that frontal lobe is used for planning and motor behavior. So when you think about where are my memories for my ability to dance the Macarena, that has to be in your frontal lobe, because the Macarena is a dance, and dance steps are motor behavior, so it must be stored in the frontal lobe. You could ask the same question about, now that my hand is dry and I'm just sitting at a laptop, can I imagine what water feels like? What warm water coming out of a spigot feels like? Well, yeah, that's easy. Well, where is that memory in my brain? Well, it's a guarantee it's in your parietal lobe. Why? Because the parietal lobe controls your ability to detect touch or other types of touch stimuli like temperature and pressure, as well as the feeling of things like water or other objects. And so the memory for it will actually be stored in the parietal lobe. Your occipital lobe controls vision, so all of your memories for anything that is visual in nature has to be stored in your occipital lobe. And of course, your temporal lobe is going to control all of your memories for hearing, for taste, for smell. So in each of these lobes, there are going to be two things called, one called a primary and two called an association cortex. So when I mention where are the memories stored, it's always the association cortex of the lobe itself. The primary section is really the initial place where input comes into a lobe. So if you imagine the occipital lobe as a vicinity, think of it just generically as an oval. In the very center of the oval, if you drew a dark black line, that dark black line in the very center of the oval would be the primary cortex for the occipital lobe. And that's the first place input comes into the occipital lobe. And then it gets analyzed and the memories for vision get stored in all the remaining section of that oval. That's essentially how it works for every one of the four lobes. Except, of course, with output in the frontal lobe, you have all of your memories for motor behavior like riding a bicycle and any kind of coordinated activity like dance steps, your ability to run down steps really fast or ride a skateboard, all stored in the association cortex of the frontal lobe. And your primary cortex for the frontal lobe is going to be the last place that signals are going to be sent before they get sent out to muscles to control your behavior. So of course, with a sensory input, the primary cortex is going to be the first place signals reach. In an output lobe, like the frontal lobe, the primary cortex is going to be the last place the information goes before it gets sent out to your muscle. Here's an image of your brain with a color-coded set of lobes on it. Now, first, if you take a look at the bottom right portion of the image, the brain stem and the cerebellum are there. Neither one of them is a lobe. Those are just identifying the sections of the brain. Those are always in the back of the brain as well. So if you're not familiar with looking at the brain, it will always be that when you have a brain stem coming out of the base of a brain, it always points in the back, kind of like a tail. In fact, the brain stem sort of looks like a tail, and of course, a tail on an animal is always in the back, and that might be one simple mnemonic way to recognize the front of the brain from the back of the brain. The brain stem is like a tail, and it always moves toward the back. Now, if we go to the opposite side and take a look at the frontal lobe, the frontal lobe is, of course, called the frontal lobe because it's in the front, and that's exactly where it is. It's one of the larger lobes overall. Essentially, that should make sense because it involves all of the memories for any particular motor behavior you might exhibit. Behind it, at basically about the crown of your head, is the parietal lobe, which does mostly touch. Behind that, all the way in the back of your brain, is the occipital lobe, which is doing processing mostly for vision. And then your temporal lobe, which has gotten its name because it literally runs next to your temples. And of course, it's right along the side of your head where your ears are, and that makes it actually very easy to remember that the temporal lobe is analyzing your hearing. Let's take a look at this question. Which of the following lobes is not used for sensory evaluation? And of course, the correct answer there is the frontal lobe. Just checking your knowledge here, occipital, temporal, and parietal, all related to your 6 sensory inputs. And then the one motor output lobe is in the front. I had mentioned a little bit about memory location, and we're going to take a look a little bit more about this and kind of expand on how the memory system is laid out. In terms of memory location, it's clear that your memories are going to be stored in the various lobe that analyzes that particular sensory input. So when you're talking about vision, your ability to remember what the inside of your apartment looks like has to be stored in your occipital lobe because your occipital lobe analyzes vision. And so all visual memories will actually be stored there. And that's going to be true throughout the brain based on the various sensory inputs in the parietal, occipital, and temporal lobes. Now, one of the other aspects of the memory system in general that will come back to us many times over in the semester is the hippocampus. The hippocampus, just like limbic system, when I say limbic system, you should automatically think emotional state. The hippocampus is always associated with memory. And in fact, the hippocampus is actually at the very end of the limbic system. So as I mentioned that the limbic system is shaped like a pair of ram's horns that start off in the very center of your brain at the thalamus, and then on both the left and right side of the head, loop around. The very tip of that semicircular horn that we've been talking about would have a section right on the very end of it called the hippocampus. And that hippocampus is related to memory formation. So as you might imagine, because the hippocampus is connected to the limbic system, emotions and memories should be very closely related together. And in fact, they are. Emotional states do have a strong influence on the degree to which you can remember something, and that story will come back to us over and over again throughout the semester. Now, memories themselves, back in the 1950s, which is just about the first point where researchers could actually start to investigate the underlying basis of the physical components of the brain and how it influenced behavior. The original theories about memory were that they might have been tiny sections of your brain, or that they were maybe a molecule that's formed through an experience. So as we talked about how amino acids are strung together to form a protein, back in the 1950s it was theorized that a memory might be formed through processing amino acids to form one large molecule that would kind of be read as a ticker tape, and you would understand a memory just by reading through this giant molecule. But it turns out none of that is actually true. Memories are not existing in single locations in your brain. In fact, when we talk about memories, you're always having to draw from separate lobes in the brain. Like, for example, think about somebody you know, like your best friend. Your best friend has a face, and they also have a voice. Well, when you think about your best friend talking to you, that just seems like one basic memory, except it's not. The visual component of your friend's face is stored in the occipital lobe, and the sound of their voice is actually stored in your temporal lobe. So when you think about what your friend looks like and how their voice sounds, you're actually drawing from two separate areas of the brain that are actually reasonably far from each other. And how do you do that? Well, it's actually the hippocampus that sets it up. The hippocampus integrates where memories are formed and links them together. so that when you want to recall a memory, the hippocampus has to be activated to find the proper face in the occipital lobe and the proper sounds from the temporal lobe, and then pull them together and then route them over to your language center so that you can express what your friend looks like and sounds like. Ultimately, I have not talked about the language center, but that part of it's going to become very important because it mostly is kind of the end result of the storage of all these memories. Once you have the memory in your head, you have to get it out. How do you get it out of your head? Well, it's a verbal report, just like we talked about in Chapter 1. You might have thoughts and feelings in your head existing in and of themselves, but we don't have any current technology to be able to immediately draw it out. The only way to really draw it out is a verbal report. So you're always going to have to link your memories to your language system before you can actually exhibit overt behavior to express the memory in language. So memories are actually very large pathways that exist in your brain, and they get activated in different ways based upon different stimuli. The memory that's formed can be activated and drawn out based upon a wide range of stimuli, but each of those different stimuli come through separate pathways into the brain and therefore may draw on the memory in slightly different ways. So one of the things we'll see when we get to the memory chapter is that the manner in which you're cued in to remember something actually influences the manner in which you remember the memory. So it's not like the memory is kind of a gold standard and it's always correct and it's always there to be accessed. It's actually accessed differently depending upon the sensory input that goes in to receive it. And so you may wind up with slight variations of a memory depending upon whether or not it was cued in from you reading something, hearing something, or seeing something. That's a broader issue for the memory chapter, but it's part of the basics of how the brain is actually designed. So when you recall a memory or retrieve a memory, which is the more technical term for just remembering, when you say memory retrieval, you can only start the access of a memory if you are stimulated by some external stimulus. You do not do anything independently. You don't have a capacity to self-generate a memory. All memories start from an external stimulus which runs through your sensory input through a sensory input pathway and then activates the hippocampus and the relevant lobe in your brain in a particular way so that that information can be pulled out. Everything always starts with a stimulus. So the hippocampus has to access the various lobes where the information may actually be, depending upon what's actually happening. And of course, your memories can be as complete as the full range of 6 sensory inputs that you can experience, right? From touch and vision and hearing, taste and smell, balance. You have only 6 actual experiences. And those 6 can be mixed up in a variety of different ways. You could be extremely excited at a Justin Bieber concert where there's lots of loud music and a lot of activity and all of this stuff going on, and all the various components of it can only be analyzed through 6 separate ways. And those 6 ways, those 6 sensory inputs, are going to be accessed by the hippocampus, which will identify where those memories or the pieces of the memories actually exist. So let's look at this question. When you remember that hard pretzels are brown, salty, and crunchy, you're drawing on the same area of the brain for the full memory of that event. Is that true or false? The answer there is B, false. You are not drawing on the same area of the brain for that memory. Brown is a visual component. It must be stored in the occipital lobe. Salty is gustation or taste. That's stored in your temporal lobe. Crunchy is a texture or a touch aspect, and that will be stored in your parietal lobe. So for a simple memory, like remembering what a hard pretzel is, what it looks like, what it tastes like, and how it feels, you actually have to access 3 separate lobes all at one time to pull the full memory of a hard pretzel together. And that happens all the time for all of your memories, very consistently in a realistic sense. Your memories are always going to involve multiple lobes, and so you're always going to be needing the hippocampus to locate the various pieces that you need. Now, the language center, I alluded to it in that for you to get information that's stored in your brain that physically exists there as a memory in your occipital lobe or your temporal lobe, but it just sits there idle, not doing anything. When you access that memory, you have to get it out. If you can't get it out of your head, there's not going to be any overt behavior, and therefore there's not going to be any measure of psychology in that case because you don't respond. So the language center is going to be a very important aspect in terms of understanding human consciousness, because most of the questions about consciousness are going to involve language as the primary source of data in how the internal experiences of an individual are drawn out of their head so that other people can talk about them and measure them. Now, for 90% of the human population, our language center is in our left hemisphere. For the remaining 10%, it's simply on the right-hand side. There's nothing wrong or special about having your language center on the right side of your brain. It's simply a variation of how biology sets up the structure of your brain. There has not yet been any recognized relevant difference in that case. But your language center is going to decode all of the sensory input of a message and then control your ability to express yourself, and which is going to involve a lot of complicated activity involving the muscles of your lip and your mouth and your throat and your lungs all in a coordinated fashion so that you can speak. Or also in a coordinated fashion, your ability to type or to write in cursive or to use sign language or any other communicative process. It doesn't matter which process it is really, you can always get the information out, but the language center is going to be primarily involved in getting that information out. Now, typically in a general psychology course, there are 2 areas of the language center that were identified back in the 1800s that kind of led to the beginning of the recognition of where the language center was, or even the fact that we had a language center. And those 2 areas are called Broca's area and Wernicke's area. Broca's area is an area that produces language production. Language production, that is, you making language, you speaking. For your ability to speak, speaking is a motor behavior, and the language center is on the left-hand side of your head. So Broca's area must be in the frontal lobe on the left side of your head, right? Because we're going to stick with the standard story. We're going to call ourselves one of the 90% of the human population that has the language center in the left hemisphere. And Broca's area is the section of the language center that allows us to make speech. Making speech is behavior. Behavior is only in the frontal lobe. Right? The other 3 lobes all do sensory input. Only the frontal lobe does output. So therefore, Broca's area must be in the left hemisphere in the frontal lobe. Wernicke's area does language understanding. Now, understanding refers to your ability to analyze sounds. So someone says something to you, and you have to analyze the noise that came out of their head and turn it into language so that you would know how to respond. Your ability to detect those sounds and to decode them in a useful way is all related to your ability to hear. And if your language center is in the left-hand side of your head, but you're talking about decoding auditory information, then you must be in the temporal lobe. That tells us something about the language center. The language center itself is not a lobe, but it is a series of functional cells that run from the temporal lobe up to the frontal lobe, because language involves your ability to hear sounds, interpret them, and then respond back. So hearing and interpreting involves the sensory analysis in the temporal lobe, and then your ability to speak back to the individual requires motor behavior, so that must be up in the frontal lobe. So your language center on the left-hand side of your head stretches from the left portion of your frontal lobe back to the left portion of your temporal lobe. Now, essentially where we're going to be going here to finish up, we're going to talk about a somewhat practical process that comes out of the understanding of a brain-damaged individual. The story of split brains. Split brains is a process that started back in the 1960s from medical doctors who were working with individuals that had severe epilepsy. In fact, these individuals were not treated well with medication or any other available tool that they had back in the 1960s, and their epilepsy was so bad that it was likely that the individual was going to die. So in a last chance attempt to save a person's life, the medical doctors had decided that they would split the person's brain down the very center. So that way, if an epileptic fit started on one side of the brain, it could no longer cross over to the opposite side of the brain and make the seizures very strong. Obviously, this is a permanent, irrevocable process that's going to take place. And so there was a lot of ethical questions about it, but because you were talking about just saving a person's life, the medical doctors went ahead with it. Once individuals with their brain split in half were available to be studied, psychologists started to investigate how they would think, because if both your left and your right hand sides of your brain are operating independently of each other, you might wind up seeing some interesting effect. And that's essentially what had happened. This analysis of split brains led to an understanding of how the left and the right hand sides of the brain, or the left and right hemispheres, work together through a strip of tissue, which is all axons, called the corpus callosum, that links the left and the right hand sides of the brain together. And so in this movie, we're going to see a description of how thinking is a little bit different in these individuals by watching a person who has actually had their brain split.
We began our journey into the human brain here on the campus of Dartmouth College in New Hampshire. I'd come to meet one of the world's leading brain scientists, Mike Gazzaniga, and a man he's worked with for over a decade. A man with two brains. You've been working a long time with Dr. Gazzaniga?
14 or 15 years.
It doesn't seem like that long, does it?
The collaboration began when Joe had surgery.
And you had this procedure to correct an epileptic problem,
Yes.
Is that right?
Try to stop the seizures. I was having seizures like every day or so, or sometimes 2 or 3 a day.
To control Joe's epileptic seizures, a surgeon severed the connection between the two halves of his brain. Cutting the corpus callosum like this prevented the spread of the electric storms that caused his seizures. But it also prevented the left and right halves of his brain from communicating with each other. In the years since the operation, Joe's epilepsy has been under control. He now earns a living at an egg farm, and in his everyday life, he's largely unaffected by the fact that his left and his right brains work independently.
Do you feel any different when you think about something than you did, any differently from the way you felt before the procedure?
It's just got a backup brain, that's all.
That's something everybody could use, right?
I found out how true that was right away when I was asked to draw a different shape with each hand. In a brain like mine, roughly speaking normal, at least all in one piece, the left half of my brain controls the right side of my body, while the right brain controls the left side.
Oh, no.
But because the two halves are connected,
Nothing wrong with that.
Getting each hand to work independently isn't easy.
Well, we were saying that the fact that Alan's hemispheres are connected, and that the motor messages from one are confusing the motor messages in the other.
I was just drawing an upside-down duck. Okay?
But when Joe is given the same task, his two hands operate as if controlled by two separate brains. What's happening is that each half of Joe's brain is given a separate instruction. He's asked to fix his eyes on the cross in the center of the screen. Anything flashed to the right of the cross goes only to his left hemisphere. Things to the left go to his right hemisphere. Because the two don't communicate, each hand does only what its half of the brain sees.
Wow, look at that. It's really like two different people doing the same.
That's right.
Same
That's
task.
that's the idea.
Okay, Joe, I want you to keep your hands out in front
In
of
an
you.
experiment that's now a classic in brain research,
Hemisphere.
Mike Gazzaniga over 30 years ago used a similar setup to find out if the two halves of the brain are specialized to do different things.
Ship.
Joe is being flashed a word only to one half of his brain. Words flashed to the right
Storm.
are seen only by his left brain, and Joe can report seeing those words just fine.
Piano.
Good.
But when a word is flashed to his right brain,
Didn't see that.
Okay. Joe, I'm going to ask you
but now watch what happens.
to draw that with your left hand.
They got me lost.
Why don't you try drawing another picture of it over here if that will help you.
Oh, phone.
Oh, okay.
Grr.
It's almost as though somebody has given him a secret communication.
That's
and
right.
now he knows that it's a telephone. But up until then, he was blind to it.
Exactly.
When Gazzaniga first did this experiment, it instantly proved that the ability to speak resides almost exclusively in the left hemisphere. Not until he sees what his right brain is drawing is Joe able to name
He
it.
said church, didn't he?
After looking at the picture.
But he had to figure it out about as long as we did. That's really interesting. It's a picture here of somebody communicating almost with another person.
And the communication is not occurring inside the head, it's occurring out on the piece of paper.
Yeah.
Blob. Really don't know.
You want to draw a little bit more?
So far, Joe has been seeing only one word.
Cat. That's a cat
Things get
sound.
even stranger when he's flashed two words, each to only one half of his brain.
The right hemisphere saw toad. and
Yeah.
And so his left hand draws a toad.
So there's the toad.
Oh, it's a toad.
And this time I was able to guess what was coming.
Will they put a little three-legged stool in there later, or what?
Joe's speaking left brain saw a stool. Saying the word lets the hand that's controlled by his right brain in on the secret.
That's great. That's really interesting. And if he had seen that with the corpus callosum intact, he would have drawn a toadstool.
Right.
Not a toad and a stool.
Right, exactly the point. I've been doing this for 35 years. Yeah.
And
It gets me every time.
Yeah, it must. It must.
This time, instead of naming the word, I want you to point to the word.
Again, Joe sees two words simultaneously. Bell goes to his non-speaking right brain. Music to his speaking left brain. When asked to point to a picture of what he saw, he chooses Bell. But when asked
Okay,
why,
why'd you pick that one?
Music.
Music?
and when asked to
Why music
explain,
up there?
It was music and bell, and it was a few minutes ago. Last time I heard any music was coming from the bells out here,
Uh-huh.
banging away.
So the bell's
Yeah.
outside here.
So, look good enough answer to me.
what's extraordinary is that Joe's speaking left brain concocts a plausible story of why he pointed to Bell, even when some of the other pictures more obviously represent music. Gazzaniga believes this determination to find cause and effect, this desire to explain, is the left hemisphere's most marvelous property.
One of the unique things to the human brain is this need to interpret why two events occurred. What was the antecedent to this? What caused this? And if you can imagine that a species like us that has that little chip in its brain that asks those questions is going to survive rather well, because it's going to figure out more about the nature of the world than a species that doesn't have that.
Okay, Joe, I want you to keep your hand.
But as I was
in front of
about to
you.
discover, the right brain has a very useful survival skill all its own.
What do you think will happen here?
So for you, we're doing a live experiment. Never done it before.
The experiment involves the 16th century Italian painter Arcimboldo, who made faces out of fruit, flowers, meat, even books. From other research, there's reason to believe that the ability to recognize faces is located exclusively in the right hemisphere. So Mike wondered if Arcimboldo's paintings would look different to each of Joe's two brains.
So will his left hemisphere say, I saw a potato, I didn't see a face? And will his right hemisphere say, I saw the face, and not comment on the fact it was made out of a potato?
You're going to see a figure followed by a choice of 2 words.
If this works, it'll be terrific, but we'll see. So here it is, live.
Okay?
The first painting goes to the right hemisphere. And Joe points to face.
Down.
The next painting goes to his left brain. And this time he points to fruits. Mike seemed pleased.
Are you having a moment?
This is too good.
Again to the right brain, and Joe sees it as a face.
Point it down.
But to the left brain,
Face made out of books.
Point it up.
What are the books?
Were you happy with what you're
Fruit.
seeing?
It's unbelievable. He's doing it. You see that?
It's good. It's going so fast. It's shifting from left to right so fast. I can't keep up with you. You're used to looking at this.
When you show him a face in the right side, the left hemisphere,
Yeah.
he's focusing in on the elements that made up the face.
Yeah.
When you show him the exact same picture in the left field, going to the right hemisphere, he's focusing on the face and not the elements.
And not the elements. If you came down from another planet and you saw faces and vegetables, you might not think there was much of a difference among them. But the brain seems to be made up in a certain way to say faces are very different from other objects.
That's right.
And one side of the brain specializes in faces. huh?
Exactly right. Exactly right. It is an adaptation that we have to detect upright faces. It's a very important. You can imagine in an evolutionary time that all of a sudden you have the ability to detect quickly an upright face. You want to read the expression on that face. You want to know if it's friend or foe. You want to have a set of questions about that face.
the right brain might be skilled at recognizing faces. But when it comes to what gives the human mind its power, the ability to reason, to invent, to interpret the world around it, Mike Gazaniga's 30 years of research has taught him which hemisphere he wouldn't want to be without.
The old phrase around our lab is, don't leave home without your left hemisphere. That's where the action is.
In this video, what we saw is that both sides of the brain do different processes. And in fact, the side with language, now generically we're going to call that the left-hand side, but remember this is a 90%, 10% aspect for the human population. The side that does language will be better able at analyzing the parts of units of things, whereas the opposite hemisphere, the right hemisphere, will more likely analyze things as an entirety but not look at the specific details of it. And there's a simple mnemonic rule to kind of keep this in mind, and that's this. Your language center is in your left-hand hemisphere, right? And that's generally how we talk about it. Now, language is a process by which you string together words in a specific format, right? So in English, the format for the words is subject, verb, and object. You cannot just scramble the subjects, verbs, and objects in any which way you want and get the same meaning. So the left hemisphere has to be involved in analyzing sequence and individual pieces for it to be able to do language. And that skill extends to all stimuli. so that your left hemisphere always analyzes things in terms of smaller units, the sequence of the units, the parts of the unit that make up the whole, like the bricks in the wall. Whereas your right hemisphere tends to look at the wall as a whole and does not notice the individual bricks that make it up. So you do have kind of different processes, but you should steer clear of thinking about an individual being a right brain person or a left brain person. That's actually kind of an old idea back from around the 1970s or 80s that really has not panned out well. That kind of idea that a person is dominated by one hemisphere versus another simply has not been supported in the research. Everybody does have left and right hemispheres that function differently, but you are not at all dominated by one versus the other. They work equally together because they are connected through the corpus callosum, which is a stretch of axons that connect together the left and right hemispheres. Now, the last slide that we have to cover is a discussion of this layout, the wiring diagram of the split brain. And this wiring diagram is actually very important in a general sense with respect to the brain, because as you will see, for you to understand and predict what behavior a person's going to exhibit requires you to understand the neural pathway of the brain itself. And it's the pathway more than anything else that will lead you to success in predicting behavior, as opposed to other things such as the neurochemistry of the brain or other aspects that we've already talked about. Pathway will dominate them all. It's not the only important thing, but it is definitely the dominating characteristic of predicting human behavior. So let's take a look at this individual in the diagram here. This person is looking at an image that has a red color and a green color on the left and right sides respectively. Now, for the way your brain is laid out in terms of vision, anything that's on your right visual field, so that is, if you're looking directly forward, anything that's toward your right, when you're not looking toward the right, but you're looking forward still, anything from your right peripheral vision comes into the pupil of your eye and crosses over to the left side of your eyeball. Take a look and follow that on the diagram. So anything in your right visual field always lands on the left side of your eyeball. And the cells on the left side of the back of your eyeball are wired to the left side of your occipital lobe in the back of your brain. The opposite is true with the left visual field. If you are looking directly forward, anything in your left peripheral view will involve light that comes into your pupil and therefore lands on the right side of the back of your eye. Again, look at the diagram on the page. The right side of both of your eyes are wired to go to the right side of your occipital lobe, which is in the back of your brain. So if a person had a split brain, that is, they did not share the information from the left side to the right side, what would this person see if you asked them had they saw the color red? Now, one bit for clarity to recognize is this pathway that you're looking at is not split during a split brain. This story is maybe a little bit more difficult, but literally above, coming out of the image, above this location where the visual system is wired, is where the corpus callosum is, where most of the information from the left and right hemispheres is shared. That portion is actually cut, and it's not drawn in this image. But the visual system is intact. That's much further down. Now, if you asked a person with a split brain, could they see the color red, what do you think their answer would be? The answer would be No. They would not be aware of seeing red in a case like this because red is in their left visual field. The left visual field will send out light that will land on the right side of each of their eyes. Those pathways from the eye back to the occipital lobe will wind up in the right hemisphere. The right hemisphere does not have language, so you cannot speak about your experiences that land in your right hemisphere. That's why in the movie that you just saw, the patient, Joe, was asked to use his left hand, because what happens is when the signals from your brain go down to control your body, everything switches over from left to right as it goes through your neck. And so the right side of your brain controls the left side of your body. The left side of your brain controls the right side of your body. So if the right side of your brain had detected red, but there's no language center over there for you to express it, you could use your left hand to point to the color red, and then you would actually show a conscious experience of having seen red. If you were asked if you see green in this scenario, the person would automatically say yes. And that's because green is on the right-hand side of the individual. It's the right visual field. The green light will come in through the pupil and land on the left-hand side of the back of their eye. Those sections on the left-hand side of the back of the eyeball are wired to the left portion of the occipital lobe, and that will be automatically connected over to the language center, which is on the left-hand side, so the person will automatically verbally express, yes, I did see the color green. But they could only use their left hand to point to a variety of option of colors as to what they've seen if the red light was processed by the right hemisphere. This sort of analysis gives you an idea of how you have to process an understanding of the circuitry of the brain to be able to predict what kinds of patterns of behavior occur. Thinking like this is especially important in terms of analyzing the types of symptoms that occur when people get brain damage. Because of your entire brain, of any one area that becomes damaged and no longer processes information through it, you wind up with symptoms that occur as a result of all of the following portions of that circuit being eliminated, and then you trying to figure out how that would actually produce a specific set of symptoms. And that concludes Chapter 2 on neuroscience and behavior.